Hello, my name is Emily Van Buren, and I'm a PhD student in the Midlars Lab. My poster is titled Evolutionary Understanding of Melanin Synthesis. Are the PO candidates in Cnidarian species more similar to humans or to insects? Melanin, as we know, is a pigment that has a variety of uses in various species. Human melanization we primarily associate with the pigmentation of our hair, eyes, and skin, but can also play a role in innate immune functions. For example, this is you can see it with the darkening of your skin during your UV radiation. Innate immune studies of melanin have also been performed in insect species such as Drosophila because it lacks an adaptive immune system. Both human and insect models have been used to understand Cnidarian systems. Chemically, we know that there are two main pathways that perform melanin synthesis and are annotated on the KEGG database. Tyrosine metabolism and melanogenesis are the two pathways, and their primary difference is the dependence on the presence of a specialized cell called melanocytes that is exclusive to melanogenesis. Humans and other mammalian species are unique because they have this specialized cell where melanization occurs. And this is the cells that you see that create your pigmentation for your hair, eyes, and your skin. Regardless of these systems, melanin synthesis is dependent on PO candidates. They're enzymes that catalyze monophenols into diphenols and kinines. These diphenols and kinines will go through non-enzymatic steps to become melanin. We see melanin as a phenotype of disease and stress in multiple studies in Cnidarian species. Eunicea Black Band disease is an example of one disease outbreak that had an unknown environmental or pathogen origin in 2011 in Eunicea corals in Florida. PO candidates have since been identified in the transcriptome and proteome of Eunicea corals inflicted with Eunicea Black Band disease. The purpose of this study is to determine what genes are present in the melanin synthesis cascade in Cnidarian species and what relevant PO candidates are present during disease and stress events. Evolutionary comparisons were done on several levels that included mRNA comparisons in evolutionary trees, Pfam domain identifications, and string network analysis through disease modeling of Eunicea Black Band. PO candidates, evolutionary trees were created using max likelihood methods in MegaX. Pfam domains were also identified for each one of the PO candidates for each of the species. And finally, string network analysis was done for Drosophila and Homo sapiens for the tyrosine metabolism, for their specific tyrosine metabolism networks, because tyrosine metabolism is not dependent on melanocytes. In our first result, the laccase PO candidate tree, we're noticing some unusual patterns of branching that are not based off of phylum, as we would assume with an evolutionary study, but in fact based off the Pfam domain associations. Specifically, we're seeing branching based off of if there's DUF218 present, copper oxidase or copper oxidase 2 present. And these are the drivers of the evolutionary tree. For our tyrosine metabolism based ones, so your hemocyanins, tyrosinase, and PPO enzymes, you see that they are truly branching based off of the phylums that are present. So, and specifically the two drivers being if it's a hemocyanin associated Pfam or if it's a tyrosinase associated Pfam. For our string networks, we're overlaying information of sequenced tyrosine metabolism genes. We see, and we're including anything that was associated with Eunicea Black Band in their transcriptome or their proteome. In the human network, we see that there's a 40% matching rate to the Eunicea Black Band disease sequence, tyrosine metabolism, and then in Drosophila, we see it at 18%. Further broken down, we see that the tyrosine degradation module is 89% matching for Eunicea Black Band to Homo sapien and 33% to Drosophila to Eunicea Black Band. Looking at this, we're seeing that the evolutionary studies and disease models are supporting that Cnidarian species are more similar to humans than to insects, regardless of this presence of a specialized cell. String networks are also indicating that Eunicea Black Band disease, while phenotypically melanized, was not actively working on melanin cascade, but is most likely in the end stages of the disease due to the high matching rate of tyrosine degradation. Laccase still needs more studies to understand how the structural composition may influence its roles in disease. Overall, we're seeing that that Cnidarian disease models can be a good representation of innate immune studies, regardless of the specialization in other species. I would like to thank Whitney Thoren, Lauren Fuse, Contessa Rickey for their work in Eunicea Black Band disease that made this project possible, and the guidance of Dr. Lauren Midlars. Thank you.